Kind of introduction. As I said, I'll be talking about chlorophyll in kelp. So where I'm beginning isn't quite where a lot of people are today. I'm a junior college student. I'm in my second year, and I've just completed majors biology. Our majors biology class ends after 2 semesters with a talk, where we do a project, and then we propose it. We give our first paper, and we get our feet in the door for getting into the field, designing and writing projects. Okay? Here, I invite you to come into the water with me and see what I found. Okay. Macrocystis pyrifera is a giant kelp. It grows to 70 meters tall in the water. It's equivalent to about 14 stories high, but it's a multicellular protease. It has no differentiating conducting tissue like phloem and xylem to really transmit these nutrients. So I found it fascinating to see why is it doing what it's doing and what will my school really let me work on with my time and my schedule. And so what I did was I designed a study to look at how this was going to happen. Giant kelp is found, Macrocystis pyrifera is found from Baja California to Northern California and Santa Cruz. It varies in color. If you look at it, you can see the color differences, and it has some attributes that really made me curious. My earlier project with one of my cohorts here was seeing if there was a chlorophyll difference in surface and deep blades, and that showed that there was a significant difference between blades that were growing on the surface, new growth blades that are about this big, versus blades that were 2 meters down and quite a bit darker. I wondered if it was temperature, I wondered if it was photosynthetic pigments, and so here I chose to look at longitudinal studies to see if northern and southern were different, and to see if maybe temperature could be a factor. As I said, my main objective was really to get some experience in knowing how to get my feet wet and, get into the field of science. You'll remember when you first had that thing that you just wanted to look at, and you wanted to bring your lab with you and go study it. And so that's what I did. I packed up a sterile lab, put it in the back of my car, and went on a road trip for 2 days and collected kelp with ice baths and preset acetone mixtures. I did a lot of collaboration with my senior technicians. I'm like, okay, what should I do? What should I do? How do I make sure that all of this stuff is going to go well? And I'm going to break the $500 organic pump. My professor told me, so prepared some backups and set on my way. As I said, one of the big factors was choosing collecting sites. I don't have a lot of data mining experience. I don't have a lot of knowledge of going through all of the literature and finding stuff, but I know how to use a map. And so I checked out Google Satellites, and in a further slide, I will tell you how I found where the kelp was. As I said, placing acetone, they need to be set in acetone because it's an optimal solvent for getting chlorophyll from the cells, from the Macrocystis. And once you remove that, you incubate it for 48 to 72 hours. And you can test it that time, you can test it a week later, and you're going to get a negligible difference in chlorophyll. And so within 72 hours at 40 degrees Celsius, I had samples that were ready to run through a special photometer and to get some results with. That was amazing. As I said, Google Maps, satellite imagery. From the top to bottom, north to south, you can see my different sites. Santa Cruz, California is up there above Monterey Bay. That San Simeon, it's right below Big Sur, really isolated population, but not population, just a beach. Santa Barbara, right by the college, you go down the stairs and you see this huge kelp forest. There's kelp all on the beach, and it's like, okay, going out. And Topanga and La Jolla were pretty easy to get to as far as the difficulties, and seeing them was the cool part, because if you zoom in, it's like over here. Let me see something. There's scales given that are showing up to show increase in magnification from 100 meters to 50 meters. And this is the general region that I would collect in. I paddle on a surfboard. and with Ziploc baggies, a mesh bag, and I'd take clippings of blades off of stipes from 1 meter down with the same blade shapes and sizes so I can get as consistent as possible, pulled them out, and dropped them last when I got back to my car. A whole bunch. It's pretty fun. Here's the results, actually. So, the data speaks fairly loudly for itself. In the south here, San Diego to Santa Barbara, the temperature is much higher, and there's a huge discrepancy between the southern blades and the northern blades. I got this, and imagine your first project showing that you're getting the results that you want, and your air pressure like this time. Oh, this is my life. And then while I was in the water, come on, like, look at this versus this. Like, this is within a day of each other. They're the same sizes, and those colors are drastically different within this one species. What I really saw from this was some future options and growth that I can go exploring. Going exploring and getting science done in the field is why I'm here and why I'm standing in front of you, little junior college student trying to get awareness. But what I found was there was a significant difference. There's a significant difference in chlorophyll between every single site, except for the 2 sites that were closest in temperature. Like 0.3 degrees Celsius difference between those 2, which is actually just a little different from that. So I feel like I want to go explore if temperature is a major factor. For now, the stats speak for themselves, the colors do, and it's really been a great experience presenting this material to my classmates and bringing it here, having the option to speak at an international conference on something kind of humble. In the future, I invite any of you to give me some advice on what kind of studies you'd like to see done and see pursued in this field, because I am an open book who's going to go to graduate school in about 2 years, so I'm always looking for programs and advisors and information. And I'd love to just go study in the environmental conditions that do select for the species differentiation. And there's chlorophyll that's changing, and there's got to be selective pressures that's really contributing to these factors. Right? So on that note, I'm sure there's plenty of time, and I hope you have a good break. But my advisors and my professors for bringing me here to Brazil, Professor Bruno and Dr. Kimo Morris are sitting in the audience today, and I want to thank them tremendously for the time that we shared together. We did a study abroad trip, and that's the reason why I'm here speaking here today. Megan Sixt was the first project with the Surface and Deep, and she is also here, along with my major professor, Dr. Tony Huntley, who could not be here today, and he's back in the States enjoying his life. But I want to say thank you. He's not enjoying it as much as we are here in Brazil, right? It's a pleasure. Other than that, I want to say thank you and open it up to any questions, please.